Hi, I'm Steph Metzger, and I'm a doctoral candidate here at the UW-Madison Dairy Science Department. I'm co-advised by Drs. Pamela Ruig and Laura Hernandez, and we work with Dr. Garrett Suin in the bacteriology department to study the milk microbiome. You'll often hear about the microbiome in terms of the dairy cow rumen. The microbiome is just the community of bacteria that live within an environment. So human skin, the dairy cow rumen, or possibly the dairy cow mammary gland. There hasn't been a whole lot of research into this previously, and people have generally looked at it in terms of, okay, let's compare at one time point clinically mastitic cows to healthy cows. So we wanted to look and see if there's a microbiome in the milk and whether this changes across lactation and how it's associated with the somatic cell count of the cow. So we sampled individual mammary quarters at Dryoff, at calving, and then throughout the first 150 days in milk. We looked at the somatic cell count of these quarters, and we cultured them in the UW Milk Quality Lab to see if they had bacterial growth. We got some healthy quarters. These had a low cell count and no bacterial growth. And then we found some quarters that had new inflammation across the dry period. So they had a low cell count before dry-off and a high cell count at calving. And then we found some chronically inflamed quarters. These had an elevated somatic cell count at dry-off and then again at calving. So these would be quarters where our dry cow therapy wasn't necessarily effective. So what we wanted to look at were two basic metrics of how we measure a microbiome. These two are called richness and diversity. The richness is the number of bacterial species in a sample, and the diversity is the evenness of these species. So if you have greater diversity, that means your environment isn't dominated by one specific type of bacteria. It's more evenly distributed. So in order to generate our richness and diversity values, we used culture-independent sequencing techniques where we can sequence the bacterial DNA that we extracted from these milk samples. So we found that these healthy quarters, which are in blue here, these had a greater richness than our inflamed quarters. So our healthy quarters have more bacterial species that we detected in this mammary gland. For all of our groups, the richness decreased between dry-off and calving. And this may be because of that dry cow therapy that we treated our cows with. It could also be due to the involution of the mammary gland. We're not really sure. And then for our diversity, the evenness of those species, the diversity didn't really change across lactation, but the diversity was greater in our healthy quarters than in our newly inflamed quarters that acquired inflammation across the dry period. So there's something going on there that we don't understand yet. Some long-term potential applications of this research would include potentially using microbiota transplants, the way you can do with a rumen, but we're not really there yet. There's a lot more research required. This is sort of the basic introduction to what's going on in this milk microbiota.